Genetics is the science that deals with the transfer of characteristics or traits from parents to offspring. So if the transfer of characteristics from one generation to other generation is same, this is heredity. For example, if the parents have black eye color and the offsprings also have black eye color, it means that the characteristics of both parents and offsprings are same. So this is heredity. And variation is different. Different traits or characteristics. For example, parents have black eye color and the offsprings have blue eye color. This is an example of variation. And both heredity and variation in genetics are controlled by genes. Genes are the sequence of nucleotides in DNA that control heredity and variation. They have the information stored in them that helps to control the traits. These two are chromosomes, one is coming from the male parent and the other is coming from the female parent. After fertilization, these two comes together in the form of pairs. These are known as homologous because both chromosomes have same gene sequence, same gene locus, same centrometric position, same chromosomal length. So they are almost similar to each other, but their alleles are different. They can be or cannot be different. You can see here that purple and yellow genes are coding for the same character or trait, that is flower color. And these genes have the same locus on both chromosomes. But here alleles are different. Purple gene have the alleles which code for purple flower color and yellow gene have the alleles which codes for yellow flower color. So alleles are the alternate form of genes. Homozygous is a genetic condition in which both alleles are same and they express same character or trait. For example, capital B or small b, these are homozygous alleles. On these chromosomes, alleles are shown with a blue color and they are expressing same flower color, that is blue flower color. So this is a homozygous condition and these alleles are homozygous alleles because both are same. Here you can see these two chromosomes having heterozygous alleles. Now both alleles are different. One allele is shown with blue color, that is coding for the blue flower color, and the other allele is shown with a purple color, that is coding for the purple flower color. In this diagram, you can see that male having black eye color. This is phenotype because phenotype is the physical character. It is the physical appearance of trait. It is what we see or what we observe. And male having heterozygous allele combination is genotype. Genotype is the genetic makeup of an individual for a particular trait. So this is heterozygous because both alleles are different and capital B allele codes for black eye color, small b allele codes for blue eye color. And here, male having black eye color cross-fertilized with female also having black eye color. And female also have heterozygous allele combination. As a result of this cross-fertilization, offsprings are produced. First offspring will have homozygous allele combination, capital B. Both alleles codes for black eye color, so phenotype is black eye color. Second allele will have heterozygous allele combination, capital B, small b. Both alleles are different. Here phenotype is black. It means that capital B allele expressed itself. Small b allele did not express. So capital B allele which expressed itself is dominant and small b is recessive. Small b will express only when it is present in homozygous recessive condition, which is in fourth case. In fourth offspring, you can see that homozygous recessive allele combination is present in which both alleles are small b, which codes for blue color. So here this offspring will have blue eye color. 3 black eye colors and 1 blue eye color is phenotype. And here genotype is 1 homozygous dominant allele, 2 heterozygous alleles, and 1 is homozygous recessive alleles is genotype. So dominant is when one allele in heterozygous condition express itself. Dominant is always represented by capital letter. And recessive is when One allele in a heterozygous condition do not express itself is recessive, and recessive is always represented by a small letter. Gregor Johann Mendel is the father of genetics. He worked on pea plant, that is Pisum sativum, in his monastery garden for 11 years and formulated two laws of inheritance, law of segregation and law of independent assortment. Now the question is why he chose pea plant? Because its flowers are hermaphrodite. It means its male and female parts are present on the same plant. It can be self-fertilized as well as cross-fertilized. Its generation gap is very short and it has 7 contrasting pairs of traits. It's diploid. Its number of chromosomes are 14. By his experiments, he concluded that genes are present in the form of pairs and they are inherited as distinct traits, one coming from each parent. Mendel's first step was formation of pure or true breeding plant. Pure or true breeding plants are those plants that have the offsprings of its own phenotype. So these offsprings are not only identical to each other but also to the parent. So pure or true breeding plants are those plants that have same offsprings. For example, if a round seed shaped plant is self-fertilized, all of its offsprings are round seed shaped. If a tall plant is self-fertilized, all of its offsprings are tall plants. So both parent and offsprings are pure or true breeding plants. Similarly, Mendel established 14 pure or true breeding plants. And then he moved on to next step. He cross-fertilized those plants which differs in only one trait. That's monohybrid cross. Capital R allele codes for round seed-shaped plant and small r allele codes for wrinkled seed-shaped plant. So he cross-fertilized true breeding round seed-shaped plant with a wrinkled seed-shaped plant. As a result, gametes are formed. One gamete have capital R allele and other kind of gamete have small r allele. After fertilization, these two alleles combine and form heterozygous allele combination, that is capital R small r. This is F1 generation. F1 is first filial generation that is formed after fertilization. Mendel got round phenotype and heterozygous genotype in F1 generation, but he was quite confused because one of the parent have wrinkled seed in parental generation but none of the offspring have any wrinkled seed. Then he decided to move on to the next step, in which he self-fertilized F1 generation having heterozygous allelic combination to get F2 generation, second filial generation. Here you can see that capital R is pairing up with capital R and small r. Similarly, small r is pairing up with capital R and small r. And phenotypically, 3 round seeds and 1 wrinkled seed appeared. So phenotype is 3:1. And genotypically, 1 is homozygous round, 2 are heterozygous round, and 1 is homozygous wrinkled. So here genotype is 1:2:1. Punnett square is a square box diagram that is used to predict all the genotypic combinations for a particular trait in breeding experiment. It is used to find out the genotype as well as phenotype. So let's find out it. Here again we are going to self-fertilize F1 generation having heterozygous alleles. So first write down the heterozygous alleles on vertical side as well as horizontal side. Capital R will multiply with capital R with small r, then small r again with capital R, and small r with small r. So you just need to multiply these alleles. And here you can see that capital R alleles code for round seeds, capital R small r for round, capital R small r heterozygous round, small r are homozygous wrinkled. Now its genotype should be 1:2:1 because one is homozygous round, two are heterozygous round, and one is homozygous wrinkled. And phenotype should be 3:1 because phenotypically 3 are round and 1 is wrinkled. So you can see here that genotype and phenotypes are different. And how we predict these genotype and phenotype by using Punnett square. Let's see how gametes are formed. Gametes are haploid cells and they have the ability to fuse with another gamete and form zygote. Let's say a trait that is coding for seed shape have the gene, and this gene have two different alleles. Capital R allele is coding for round seed shape and small r allele is coding for wrinkled seed shape. So now these alleles in a gene separate from each other and form two gametes. This is a female germ cell having two alleles capital E small e on gene in ovum, and these alleles first will separate from each other and form two gametes. One of the female gamete fuse with the male gamete that is sperm cell to form a zygote. Zygote is a diploid cell which is formed by fertilization, and this zygote have now new allele combination that is capital E capital E. Next is law of segregation. What law of segregation says? It says that two coexisting alleles for a trait in an individual separates from each other and form two gametes. Gametes are formed by meiosis and these are haploid cells. And during fertilization, male and female gametes fuse with each other to form a zygote, which is a diploid cell. I hope you like this video. Please like and subscribe FMZ Biology to get more videos like this.